Hello students, myself Dr. Mohammad Zafar Iqbal. I am from Government Arts and Science College, Karwar. And in this video, we are going to discuss about the first part of the protein biosynthesis. That is, in this video, we are going to discuss about the different components which are necessary for the protein biosynthesis. And in the first video, we already discussed about the genetic code. The understanding of the genetic code is very important for the understanding of the protein biosynthesis. And if you understand the genetic code, then only it becomes easy for you to understand the concept of the protein biosynthesis. I think most of the students know what are the proteins. The proteins are nothing but they are the very important biomolecules present in our in all the organisms, and whatever the functions are there, each and every function is carried out by means of the protein. That's why the proteins not only give the functions, they are also very important for the structure of an organism. We take so many different types of the structures we have, so many organs we have, so many tissues we have, and we have got so many different types of the cells. And if you take the architecture or the structure of these all these cells, tissues, and organs, you'll find that the proteins play a very significant role. And in this way, the proteins are one of the very important molecules. And when we discussed about the information pathway or the central dogma of molecular biology, in that we discussed that if you take the information flow, the first molecule is the DNA. From the DNA, the RNA is synthesized, and from the RNA there is a synthesis of the protein molecule, and whatever the information pathway is there, the information pathway ends at the stage of the protein. That's why the proteins are nothing but they are the end stage of the information pathway. And since they are the end stage of the information pathway, that's why they are very important for the function of an organism. That's why that how exactly they are synthesized in our body, that we are going to discuss in this video and in the next video. First, we get into this particular, we are going to discuss about some of the major questions and developments. Today, we have a lot of information about the protein biosynthesis. We have got a lot of information about the transcription. We have a lot of information about the genetic code. But if you get back to 1960 or before 1960, nobody was knowing anything about the proteins. Nobody was knowing anything about the mechanism of the synthesis of the protein, how exactly the proteins are synthesized. That's why in this video, we are going to, in this first part of this particular video, we are going to discuss the major questions which led to the identification of the protein biosynthesis. Some questions were raised, and these questions and the developments were responsible for the identification of the process of the protein biosynthesis that we are going to discuss in this video. And second important thing is we are going to discuss about the different types of the components. We know that if you take the protein biosynthesis, the protein biosynthesis is a highly complicated mechanism. So many different types of the proteins are involved, so many different types of the enzymes are involved, so many different types of the RNAs are involved, and all those we are going to discuss in this particular video. We are going to discuss about the what exactly mean by mRNA, what exactly mean by tRNA, what exactly mean by ribosomes, what are the ribosomes, and what is the significance of the ribosomes in the protein biosynthesis. And most of the students might be knowing that the site of protein biosynthesis is nothing but it is the ribosomes. If you take where exactly the protein biosynthesis, if you take the cell, so many different organelles are there. There is a nucleus. Within the nucleus, DNA is present, chromosomes are present. And if you take the cytoplasm, in the cytoplasm, so many different types of the organelles are present, like endoplasmic reticulum, Golgi complex, and ribosomes are also present in the cytoplasm. And now we have to identify where exactly the protein biosynthesis will take place. And when this particular thing was investigated, that time they found that the protein biosynthesis occurs at the ribosome. That's why the ribosomes are nothing but they are the sites where exactly the protein biosynthesis will take place. This is the very important point, and this is the main function of the ribosome. That's why the ribosomes are very significant. And one more very important point associated with the ribosome is that if you take the ribosomes are constructed only from two biomolecules. One is the RNA, second one is the protein. Apart from these two biomolecules, you don't find anything else. You don't find any carbohydrate. You don't find any kind of the lipids or the fats. And that's why whatever the ribosomes are there, the ribosomes are completely constructed from the two biomolecules. One is the RNA and second one is the protein. That are the only two biomolecules which are present in the architecture of the ribosome. Then we are going to discuss about the last, that is the amino acyl tRNA synthetase. This is an enzyme, and as the name itself indicates that amino acyl tRNA synthetase means this is the enzyme which is responsible for the charging of the amino acid. That is, the charging of the amino acid means they are going to attach to the tRNA molecule, and that particular attachment of the amino acid to the tRNA molecule is facilitated by means of one enzyme, and that enzyme is called the amino acyl tRNA synthetase. Don't get confused. Amino acyl tRNA synthetase is nothing but it is an enzyme which is responsible for attaching amino acid to a specific type of the tRNA molecule. That is the when a particular amino acid goes and binds to a tRNA, and who is going to help that particular process? That particular process will be helped by means of a one particular enzyme, and that particular enzyme is called the amino acyl tRNA synthetase. And I think you know what is the meaning of synthetase and the synthase. If you take the synthetase means it involves use of energy either by hydrolysis of the GTP or by means of the hydrolysis of the ATP. And synthase means that there is no involvement of any energy. That's why we have to be very, very cautious while using this particular. Whenever you write, you should not write amino acyl tRNA synthase. Then it is wrong. It is not a synthase, it is synthetase, because it uses the ATP hydrolysis for the energy to guide this particular reaction, to run this particular reaction. That's why this is also one of the very, very important terminology in the biochemistry and molecular biology that the student, most of the time the students neglect, but it should not be. And synthase means no use of ATP or the GTP, and synthetase means there is a use of ATP and GTP. Just by means of the name, you can identify whether this particular enzyme uses ATP or it doesn't uses the ATP. That is also one of the very, very important. These are the some of the objectives which are set for this particular video. And now we'll move to the important landmarks. And you know that when something big happens, then some important landmarks will be there, some important discoveries will be there, or some important observations are there. And one of the most important observation for the identification of the phenomena of protein biosynthesis is the question where exactly the proteins are synthesized. This is the basic question, because if you take an organism, so many different regions are there, so many different types of the organs are present, and within the organs so many different types of the tissues are present, and within the tissues so many different types of the cells are present. And even if you take one particular cell, so many different regions are present. Nucleus is present, cytoplasm is present, and within the cytoplasm so many different types of the organelles are present. And out of these organelles where exactly the protein biosynthesis will take place, that is also very, very important. That is a very, very genuine question. And in this particular, in order to address this particular question, number of studies were carried out. And one of the study which has clearly indicated that where it has clearly showed that where exactly the protein biosynthesis take place. And what they have done in this particular experiment, they have simply taken the amino acid. What type of amino acid they have taken? They have taken the radio labeled amino acid. Why they have taken the radio labeled amino acid? Because they wanted to trace the movement where exactly those particular amino acids are going to be incorporated, that they wanted to monitor. And if you use normal amino acid, then it may not be possible to trace them. And that's why they use the radio labeled amino acid, and they allowed to remain like that for some time. And at regular intervals, they have taken the tissues and they have homogenized, and they have studied where exactly what is the condition of that particular homogenate. And when the number of hours or days elapsed after the inoculation, after the introduction of this particular radio labeled amino acid into the mice, then what they observed? They observed that the radioactivity was found in case of all the regions. It was present in the cell, it was present in the tissues. Wherever you take, everywhere there was a presence of the radioactivity. It means that just by means of when days and hours elapse, then it is not possible to identify where exactly proteins will be synthesized. When they come to minutes, then they found that whatever that particular protein biosynthesis, that is the radio labeled amino acids, were only confined to the one particular region of the cell, and that particular region was identified as the ribonucleoprotein particles. They only said that that particular complex is not, that time they were not knowing anything about the ribosome. That's why they have not named it as a ribosome. Only they have said that there is a ribonucleo particles are there, and these ribonuclear particles are the places where exactly the protein biosynthesis will take place. In this way, you can find that Hoagland has. In this way, you can find that it has been completely proved that the protein biosynthesis will take place in the one particular region, and that particular region they identified it as a ribonuclear particles. And today we know that that particular region is nothing but it is the what we call it is a ribosome. That is one of the very important question. And by that, this particular question was addressed, and answer was experimentally being proved. And this is the one of the very, very important major development which is responsible for the identification of the process of the protein biosynthesis. The second question is the how the amino acids get activated, because by that time they were aware that the amino acids are present in free form. That is the 20 different amino acids are present in free form. And when you take this particular amino acid, they should get incorporated into the amino acid by means of a peptide bond. And who is going to activate this particular amino acid? That was the second question. And in order to address this particular question, what they have done, they have taken the amino acid, and when they incubated the amino acid with the ATP and the cytoplasmic plasmic fraction, what they found that the amino acid got activated, or they got charged. It means that whenever you take this particular amino acid along with the ATP plus the cytoplasmic that is the part of the cell, then what will happen in the cytoplasmic part of the cell? The nucleoprotein particles, that is the ribosomes, will be there, and whatever is the ATP is there, the ATP will also be available for the extraction of the energy. And that's why I can find out whatever the different types of the things which are necessary, even tRNAs are also present in the cytoplasm. And in this we can find out by using this particular by this particular question, the scientists were able to identify that the amino acids get charged, or they get activated by means of the enzyme, and that enzyme they have not identified at that time, and said that this is an enzyme, and that particular enzyme is responsible for attaching the amino acid to the tRNA. And whatever the tRNA is there, the tRNA is nothing but it is the one of the most important molecule which is responsible for the incorporation of the amino acid into the protein molecules. And in order to do that, what has to be done? It has to be the amino acid should get activated actually. And in order to activate that particular amino acid, it is they have identified this particular. These are the two important questions, and these two important questions, along with the genetic code, identification of the genetic code, is responsible for the that led to the discovery of the protein biosynthesis, where exactly the protein biosynthesis will take place. Now I will try to identify different components of the protein biosynthesis. We already said that there are different types of the components are involved during the process of protein biosynthesis. The first one, of course, is nothing but it is the mRNA molecule, because you have to have the which protein you are supposed to synthesize, and what type of the sequence it should be. And in order to determine, you need to have a message from the genetic material, and that genetic material is transcribed into the mRNA. And whatever the mRNA is there, the mRNA is nothing but it encodes the information for the formation of the protein molecule. That's why I found that whatever the mRNA is there, the mRNAs are called the messenger. If you take M stands for the messenger RNA. That's why what message it carries? It carries message for the formation of the protein molecule. That's why it is called the mRNA. This is the first and the most important component which is necessary for the protein biosynthesis. Second one is the tRNA. We already discussed that the tRNAs are nothing but they are the molecules which have the ability to carry the amino acid, and at the same time they also have the ability to read the information which is present on the mRNA. Otherwise nobody can read, because you know that genetic code, three nucleotides are, that is the three bases are present. A group of three bases, what we call it is a codon, is responsible for giving information for the incorporation of the amino acid. And who is going to identify what message that particular codon has? And that's why in order to do that, you can find out there is another important type of the major RNA. That particular major RNA is called the tRNA. The tRNA is so called because T stands for the transfer RNA. Since this is responsible for the transfer of the amino acid to the protein, nascent protein, that's what is called the tRNA. This is also another very, very important biomolecule. Now, if you take the third biomolecule, the third biomolecule is the rRNA, because we already know that the ribosomes are very important, and the ribosomes are the places where exactly the protein biosynthesis will take place. And if we already discussed that if you take the architecture of the ribosomes are only constructed from two biomolecule. One is the RNA, second one is the protein molecule. The RNAs which are responsible for the formation of the ribosomes, they are called the rRNA. Since they are directly responsible, they have only one function, that is to construct the ribosome. And in order to construct the ribosome, you can find that since these particular RNAs are involved, that's why these are called the ribosomal. R stands for the ribosomal RNA. This is very, very simple. C, M stands for the messenger RNA, T stands for the transfer RNA, and R stands for the, since they are part of the ribosome, that's why they are called the rRNA. This is also third important component. Now we have got three different types of the protein molecule. These are the three different types of the protein. These are the three different types of the RNA molecules, and these three RNA molecules are called the major RNAs. And why we call them as a major RNA? Because they are present in a slightly larger quantity as compared to the minor RNAs. You take the scRNA, snRNA, suRNA, miRNA, and so many different types of minor RNAs are there. They are regulatory in function. They are present in a very, very small quantity. That's why they are. It doesn't mean that doesn't mean that they don't have any significance. They are also very important. But only thing is that they are required only for the regulatory functions. As far as if you take these three RNAs, that is the mRNA, tRNA and RNA. These are the three different types of the RNAs which are the present in larger quantity. And apart from that, you can find that these three RNAs are must for the protein biosynthesis. If you have one of the RNA, take for example, you have only mRNA and you have got only tRNA, you don't have the RNA, it means that protein biosynthesis cannot take place because there is no site for the protein biosynthesis. And the same way, if you take the mRNA is present and RNA is present, but there is no tRNA. Now everything is there, message is present, the site is present where exactly the protein biosynthesis to take place, everything is there. The only thing is that the RNA molecule which has the ability to read the codon and the RNA molecule which has got an ability to bring the amino acid is not at all present. That's why it is not at all possible to synthesize the protein. And you have got tRNA as well as the RNA, but you don't have the mRNA. Then also protein is not, biosynthesis is not possible because you don't have the message to copy what amino acid is to be incorporated and what should be the length of the protein molecule. Everything is decided by means of the mRNA. That is all the that's again for that. if it, if you take these three different types of the RNAs, all these three different types of the RNAs are indispensable. And you must have these three different types of RNA, then only it is possible to synthesize the protein molecule. And if you don't have these three different types of RNA, it is not possible to synthesize the protein. The next component is we already discussed, enzyme is required, and that particular enzyme is called the amino acid tRNA synthetase. And what is the function of this particular enzyme? This particular enzyme is responsible for attaching the amino acid to the tRNA molecule. Whatever that particular process is there, attaching the tRNA, that is amino acid to the tRNA molecule, is to be carried out by an enzymatic process, and that particular enzymatic process requires ATP, and by using the energy extracted from the ATP, the amino acid tRNA will attach the amino acid to the tRNA. That is the also, that's why this is also one of the very, very important component which is the necessary for the protein biosynthesis. Now we'll discuss about the mRNA. What are mRNAs? We already said that the mRNA, M stand for the messenger RNA, and these are important molecules because they encode the information for the protein biosynthesis. Yes, any other RNA, all RNAs are synthesized by means of an enzyme called the RNA polymerases. We know that there is an enzyme, and this particular enzyme is also responsible for the condensation reaction. And this enzyme is responsible for copying the RNA, for synthesizing the RNA. That's why it is called the RNA polymerase. This is the very, very important enzyme which is necessary for the protein biosynthesis. And then the second important thing is the 4 nucleotides. 4 nucleotides necessary because they are the raw materials. That is the ribonucleoside triphosphates are necessary. Yeah, that is the ATP, GTP, UTP, and CTP. These 4 different types of the nucleotides are also necessary. That's why this is the second very, very important type of the component. The third important component is the template is required because you have to copy the mRNA gets copied from the particular template DNA, and that's why I can find out the template DNA is also very important. This is the third very, very important thing which is necessary for the synthesis of the not only mRNA, all mRNAs. Just I'm trying to highlight the mRNA. We are going to discuss this particular thing once, and whatever we are discussing, all these things are applicable to all the different types of the RNAs also. That's why this is the another thing. Then you can find that there is no need of the primer. This is very important. And when you discuss about the replication of the DNA molecule, that time we discussed that there is a compulsory need for the that is the energy that is the primers. And if the primers are present, if there is a free hydroxyl group is present, then only the DNA polymerase can only add nucleotides to the existing chain. It means it will only add nucleotides to the already existing chain. That's the primers are required for the DNA synthesis. But in case of the RNAs, you can find that there is no need for the primer. The RNA polymerases have the ability to synthesize the RNA from the first position itself. That's why there is no need of any primer. This is also very important. Then the primary transcript. What is primary transcript? The primary transcript is nothing but once the mRNA is synthesized, once the mRNA or any other tRNA is synthesized, it is always present in a one particular immature form, and that particular immature form is called the primary transcript. And whatever the primary transcript is there, primary transcript has got information not only that is it has got some sequences which has to be removed. It has to be removed, then only it will become the mature RNA. That's why whatever this particular thing is, this is called the primary transcript, and the primary transcript get converted into another. It will undergo some post-transcriptional modifications. And what are the post-transcriptional modification? Like that is the removal of the introns, because if you take the primary transcript, it has got introns as well as the exons. And in order to remove that particular introns, because exons have the information for the amino acid, they give direct message for the formation of the protein molecule, but introns do not have that particular message. That's why the trimming is required, and that particular process of removal of the introns is called as the splicing. We come across one particular term called as the splicing, and the splicing is very important. The splicing is responsible, and it is actually responsible for removal of the unwanted, that is the introns are removed, and other different types of the post transcription modification will also take place. That's why this is also another very important thing which occurs during the synthesis of the thing. And after the post transcriptional modification, the primary transcript gets converted into the mature mRNA molecule. This is another very important point. That's why now whatever mature mRNA is there, this is the mature mRNA molecule will have message for the formation of the protein molecule. Now exactly there's a reading frame will be there. A group of 3 nucleotides will give indication for the for a particular amino acid. In this, you can find that whatever the mature mRNAs are there, the mature mRNAs will have certain signal sequences, and apart from the signal sequences, the mature molecule will have the codons, and that codons will give message for a particular type of the amino acid. And apart from that, you can find that there is the initiator codon, that is the start codon, and there is a termination stop codons will be there. And these are the some of the things which are necessary for the mature mRNA. This, I think, we have understood what exactly the process of the mRNA synthesis, RNA synthesis will take place. Now, next is the important thing is there is some of the, we are going to discuss some of the important type of the modifications which are there in case of the eukaryotic mRNA. And the first one is the fifth end structure. What type of the fifth end? If you take the mRNA molecule, it will have got a fifth end and it will have a third end. And what type of the structure is present in the fifth end, that we are going to discuss. And at the fifth end, you can find that in case of most of the eukaryotic mRNA, you will find that there is a capping is present. And this particular capping is done by means of a 7-methylguanosine residue at the fifth terminus. And this has got a very unusual bond, and that particular bond is called the 5' triphosphate linkage. This is very unusual bond. And whatever this bond is there, it is occurring between the fifth end and the fifth end. That's why it is called the fifth triphosphate linkage. And whatever this particular linkage is there, that is responsible for the addition of one particular methylated group at the guanosine at the 7th position. That's why it is called the 7-methylguanosine residue will be added, and this is going to work as the cap for the fifth end. That is the that is going to become the cap of this particular fifth end. And what is the function of this particular cap? This particular cap is mainly responsible. It is responsible for two important functions. One of the very important function is the it protects the newly synthesized mRNA from the degradation. So many enzymes are there, ribonucleases are there, and these ribonucleases can damage the mRNA molecule. And in order to protect that particular mRNA molecule, you will find that this particular cap is required. And since it has got a 5' triphosphate linkage, that's why it will not be, it is not easy for the enzymes to break that particular bond. That's why you can find that the fifth end gets completely protected. That's why exonuclease like activity is not at all possible at this particular thing. The second important function of the fifth end cap is the cap, or the structure is it helps to bind with the ribosome, because we know that the ribosome should get bounded to the mRNA, that is the ribosome. And in order to attach this particular mRNA to the ribosome, there is a need for this particular fifth cap. The fifth cap is going to assist. It's a very long mechanism is there that we may discuss in some other class, in some other video. But at this particular point of time, just remember that the ribosome, the fifth cap at the fifth end, is responsible for binding up of the mRNA to the ribosome. This is also very important, because the mRNA, where exactly the protein biosynthesis is going to happen, the protein biosynthesis is going to happen in the ribosome. And that's why, since the protein biosynthesis is going to happen in the ribosome, that's why I can find that this particular part here exactly the mRNA should be placed. And in order to place that particular mRNA at the ribosome, you can find that this particular cap plays a very significant role. That is also another important function. Then what is the condition of the third end structure? If you take the third end of the mRNA molecule, in case of the eukaryotes, you will find that there is a poly A tail is present. And why it is called as a poly A tail? A stands for the adenine, and since there is a long addition of the adenine, chain of adenine will be there, and this particular long chain can have somewhere about 80 to 250, depending upon the how many days that particular mRNA is to be retained in the cell. And if it is to be retained and immediately to be used, then you can find that it will have a 80 or 90 adenines will be there at the third end. And if that particular mRNA is to be kept for a very, very long time, if you take example of in case of the RBCs, and whatever the RBCs are there, they do not have any nucleus, it means that there is no possibility of the transcription. And that's why there is a preformed mRNAs will be present in the RBC. And whatever this particular RBC, if you take the that is the lifespan of RBC, it is 120 days. And it means that whatever the mRNAs which are deposited in the RBC have to be retained for about somewhere about 120 days. That's why you can find that in such type of the condition, you can find that the length of the poly A tail can be more than 100. That is, sometimes you can find it can be somewhere about 250 number of adenines will be added. And what is the function of this particular poly A tail? Again, this is a most important structure which is responsible for protecting the RNA from that is the degradation. Whatever the enzymes are there, degraded enzymes are there, these enzymes try to convert the mRNA into the mononucleotides, and that has to be protected. If it is done, then there's no use of the this thing. Proteins cannot be synthesized. That's why you find that in order to protect that particular DNA from the third end, this particular poly A tail will be there, and the number of the poly A, that is the length or the size of the poly A, can vary from 80 to 250. That is the sometimes you may only get a 80 number of poly A's, and in some others mRNAs there may be possibility of 122, and some other mRNAs there may possibility of 200 or 250. Altogether you can find that whatever the size of the poly is there, it can be anywhere between 80 to 250. 250. This is the very, very important thing to remember. Next, we will move to the tRNAs. This is the tRNA molecule. Now, if you take this particular tRNA molecule, you can see that there is a. This is also RNA means it is also single-stranded. But only thing is that because of intramolecular, there is a intramolecular hydrogen bonding is there. That's why in some regions of this particular tRNA molecule, you can see double-stranded structure, double-stranded like structure. This is because of the intramolecular binding. Here you can find that this is the fifth end. This is the third end. From here, this particular RNA molecule will get start, and the total it will reach up to this particular point. This is going to become the third end of that particular that is the tRNA molecule. And this is the structure which is written on the paper. But if you actually see the three-dimensional conformation of this particular tRNA, it will be like this inverted L shape. It is called as the clover leaf shape, or we can say that inverted twisted L shape can also be said. And whatever this is the exact structure which is present in case of the cell. That's why it will have be, if you take the three-dimensional structure, in the three-dimensional structure it looks like this. Now we'll try to understand some of the basic things associated with the tRNA. The tRNAs are called as the adapter molecules. Why they are called as the adapter molecules? Because we already discussed in one of the slides that the tRNAs are responsible for two different types of the function. One is they have the ability to bind to the amino acid. That is, they have the ability to bind with the amino acid. That is one function done by one particular arm, that is called the amino acid arm. And there is another function that is to read the codon actually. And in order to read the codon, you can find that the tRNAs has got an anticodon. By means of a Watson and Crick base pairing, the anticodons will interact with the codon, and if the proper hydrogen bonding occurs, then it means that this is the correct tRNA of the codon. And if the mismatch occurs, then automatically you will find that it will get expelled, and the new tRNA will come and it will try its luck. And that's why, since the tRNA molecules have got a multiple function, that's why they are called as the adapter molecules. How we use adapters for the electrical appliances. In the same way, you can find that adapter molecules are nothing but we can use, they have a multiple function, and since they have got a multiple function, that's why they are called as the adapter molecule. And when you take the mRNA has got only one message, that is to encode the protein. Thus, if you take the RNA has got only one function, that is to become part of the ribosome. But if you take this particular tRNA, you will find that here there are two important functions are there. They have the ability to attach to the amino acid and carry it to the site of the protein biosynthesis. Second important thing is that they are the RNA molecules which have the ability to read the message of the mRNA. That's why they are got two very important functions. And they are generally synthesized as a long precursor. They are not synthesized individually, because if you take the size of the tRNA molecule, they are somewhere about 73 to 93 bases. And since they are very small molecule, that's why you find that they are not synthesized independently. They are synthesized in a very, very long precursor. Post-transcriptional processing will take place, and that particular post-transcriptional processing is responsible for the release of the individual tRNA molecules. And I already said that this particular structure, if you take this particular structure, this is called the clover leaf structure. It has got a if you take the three-dimensional structure, it looks like a clover leaf, or it looks like a inverted twisted L. That's why it is called the clover leaf structure. This is another important thing. Then if you take this particular molecular weight, what is the molecular weight of this particular tRNAs? The molecular weight of the tRNA molecules vary, because we have got at least 32 different type of the tRNAs in our body. That's why we can't give one particular number. That's why the relative molecular weight can be anywhere between 24,000 to 31,000. This is the molecular weight which is there for the tRNA molecule. Some of the tRNAs can have a molecular weight of about 24,000. Some of the tRNAs can have a molecular weight of about 30,000. And in this you can find that whatever the all different types of the tRNAs are there, all these tRNAs have a molecular weight between 24,000 to 31,000. This is also another very important point to remember. And sometimes it is asked in the objective type questions, or in case of some of the competitive exam. That's why it is important. Now, if you take the total number of the bases, as already said that the total number of bases in case of the tRNAs varies from the 73 to 93. Altogether, you can find that some of the tRNAs can have a basis of about 73, and some of the tRNAs can have a basis of about 93. Altogether, you can find that if you take all the different types of the tRNA molecules, they can have any basis in between 73 to 93 bases can be present. This is the very important point to remember. Then you can find that there are 4 arms are present. Now, if you observe this particular structure, you can see that this is the one arm, this is the second arm, this is the third arm, and this is the fourth arm. Altogether, you can find that there are 4 arms are present. This particular arm is called the amino acyl arm. Why? Because this is the arm where exactly the amino acid will attach to this adenine, and in this particular, since the amino acid is going to be attached at this particular region, that's why this particular arm is mainly responsible for attaching and carrying the amino acid to the ribosome. That's why this particular arm is called the amino acyl arm. It is also called the acceptor stem, acceptor arm. That's the most important term is the amino acyl arm. That is better to use instead of using this particular terminology. That's why it is better to call it as a amino acyl arm, because this particular arm is responsible for attaching and carrying the amino acid. That's what is called the amino acyl arm. Exactly opposite to this particular region, you will find that there is one more arm is present, and this particular arm is called the what we call it is this is called the anticodon arm. Why we call it is anticodon arm? Because this is the codon, this is the arm of the tRNA molecule which has got ability to interact with the codon. And now, if you observe this particular region here, you can find out that 3 bases are there, A, and G. And whatever these bases are there, these 3 bases are called the anticodon. These anticodons will interact with the codon of the mRNA molecule. And if the that is the Watson and Crick base pairing takes place, then it will it is a correct tRNA molecule. If there is a mismatch occurs between the anticodon and the codon, then it means that this is the wrong tRNA. That's why it will be expelled. That's why, since this particular arm is responsible for reading the message of the codon, that's why this is called the anticodon arm, or it is called the anticodon loop. Then this is called the D loop. Why it is called the D loop? Because it has got some unusual bases, that is the dihydroxyuridine. And because of the presence of the dihydroxyuridine, you can find that this particular arm is called the D loop. And one more important arm is that this is called the T pseudouracil C loop. And whatever this particular loop is there, this is also very important loop. And here, why it is called the T ribothiamidine T pseudouracil loop is that it has got one particular very unusual base, and that particular base is called the pseudouracil. Here you can see that this is one base called the uracil. In that they have drawn a line. This is the pseudouracil, and because of the presence of the pseudouracil, that's why it is called the T pseudouracil C loop, or the arm. And what is the function of these 2 arms? We already discussed about the function of this arm. We already discussed about the function of this arm, and now we have to see what is the function of D loop and the that is the ribothiamidine pseudouracil C loop. That we have to understand. Whatever these 2 loops are there, these 2 loops are very important for structural conformation. As we already said that it will attain this type of the conformation, and in order to facilitate the conversion of this type of the that is the mRNA molecule, single-stranded tRNA molecule into this particular 3-dimensional structure, these 2 loops play a very significant role. That's why if you take these two loops, you can find that these are the loops which are responsible for the conformation, 3-dimensional conformation of the tRNA molecule. Now, if you observe this particular tRNA molecule, what you are going to see? You are going to see that some of the bases are highlighted like this. or here you take all these things. Why they are highlighted? Because they are called as the modified bases. They are not normal bases. As in case of AT and AU and GC, they are not normal bases. They are modified bases. Here you can find that adenine is methylated at position 1. Here you can find that this is the pseudouracil. Here this is called the ribothiamidine. Thiamine should not be there. But in case of the tRNAs, you will get a ribothiamidine will be there. Here you can find that there is a guanine is present. It is methylated at 7th position. Then you can find that this is the cytosine methylated at 5th position. And in this way, you can find that if you observe this particular thing, you can find that if you come to this particular region, these are the two, that is the dihydroxyuridines are present. And altogether, you can find that if you take any tRNA molecule, minimum of 8 modified bases will be there. That is also one of the very important thing. And if you take the first position itself, that is if you take this particular, this is called the guanosine, the first nucleic base of the tRNA will always be guanine, and it is all phosphorylated actually. And now you can find that if you take this particular structure, you can find that all together, if you take the complete anywhere between 73 out of 73 to 93 bases, somewhere about 8 to 10 bases are modified. And one more important point is if you take this particular region, that is called the CCA, and it is highlighted in some different color. Why? Because this particular region is also very important. Why it is important? Because this is the place where exactly the amino acid is going to be attached. Since the amino acid is going to be attached at this particular adenine, that's why this particular region is the acceptor, that is the triplets, and whatever these acceptor codons are there, acceptor nuclear bases are there, at the adenine region, you can find that there is a attachment of the amino acid to the tRNA. That's why this is also important. I think we are able to cover some of the important points related to this particular tRNA molecule. I think this much information is okay. That already said that guanine is phosphorylated at the 5th end, and if you go to the next point, that is the CCA at the 3rd end, and already said that out of this you can find that whatever this adenine is there, this is the place where exactly the amino acid is attached, and that's why this particular CCA end is also very important, because this is the place where exactly the charging of the amino acid will take place. That's why this is also important. These are some of the basic things which we are covering for the tRNA. And if you want more details, then we have to discuss independently. That we are going to do in some other video. In this particular context, I hope this much information is enough for the students to understand. Next is the protein. Next important component of the protein biosynthesis is the ribosomes. We already said that the ribosomes are nothing but they are the places where exactly the protein biosynthesis will take place. It means that they are the sites of the protein biosynthesis. And if you take this particular site of the protein biosynthesis, you can find that the ribosomes are present in. If you take the holoribosome, it is we have to have two different categories. One is called the prokaryotic ribosome. Second category is called the eukaryotic ribosome. That's why we are trying to understand independently, because they are sharply different from each other. That's why first we will discuss about the prokaryotic ribosome. Then we will move to the eukaryotic ribosome. In this slide, we are going to discuss about the prokaryotic ribosome. And if you take this particular ribosome, this is the holoribosome, and if you take the sedimentation coefficient, it has got a 70s. It is called the 70s. And whenever you take this particular 70s, it gets dissociated into two fractions. One of the fraction is called the 50s, and second fraction is called the 30s. That is the 30s and the 70s. It means that 70s gets dissociated into two different components. One of the component is smaller in size. That particular component is called the small subunit of the ribosome, and one of the component is larger in size. That's what is called the larger subunit of the ribosome. In this way, we can find that this is the basic structure of the prokaryotic ribosome. If you take the prokaryotic ribosome, the prokaryotic ribosome will be like this. It will be hollow. Ribosome will have both the small, that is the small subunit of the ribosome, and it has got a large subunit of the ribosome. And once it undergoes dissociation, then you can find that it gets converted into two different components. One is called the 30s, that is the small subunit of the ribosome, and second is called the large subunit of the ribosome. The large subunit of the ribosome has got it is called the 50s, and small subunit is called the 30s. This is some important point. Now we will discuss about the what type of the, since already we have said that the ribosomes are constructed only from the two biomolecule. One is the RNA, second one is the protein molecule. The RNAs which participate in the construction of the ribosome, they are called the rRNAs. Since they are part of the ribosome, that's why they are called the ribosomal RNAs. And if you take the ribosomal RNAs, in case of the large subunit of the ribosome, you can find that there are two important RNAs are there. One is called the 5s rRNA, and second one is called the 23s RNA. And if you take this particular small, that is the 5s RNA, it has got somewhere about 120 nuclear bases are present. And if you take the 23s RNA, you will find that there are somewhere about 3,200 that is the bases are present. Altogether, you can find that if you take the 5s RNA, 120 bases are present. In case of the 23s, you can find that there are 3,200 that is the bases are present. And there are 36 different types of the proteins are present in case of the small subunit, large subunit of the ribosome. Altogether, you will find that if you take the large subunit of the ribosome, in the large subunit of ribosome, there are two different types of the RNAs are present. One of the RNA is called the 5s RNA, and second RNA is called the 23s RNA. And if you take the 5s RNA, it has got 120 bases are present. And in case of the 23s RNA, there are 3,200 bases are present. This is the detail with this thing. And how many different types of proteins are there? 36 different types of proteins are present in this particular large subunit of the ribosome. Then if you come to the small subunit of the ribosome, the small subunit of the ribosome has got only one type of, if you take this 30s ribosome, that is the small subunit of the ribosome, it has got only one type of the rRNA, that is called the 16s RNA. And if you take the 16s RNA has got somewhere about 1,540 bases. Whatever figures we are giving, these are the figures for the E. coli. We are taking an example of the E. coli. And in the E. coli, we are discussing, that's why in front of, if you take the 16s RNA, the 16s RNA has got a basis of about 1,540 bases are present. And how many number of proteins are present? If you take the different types of the proteins, 21 different types of the proteins are present. This is the some information related to the large subunit of the ribosome and small subunit of the ribosome. Now, another important point which I forgot to mention is the size of this particular thing. If you take this particular holoribosome, it has got a molecular weight of about 1.8 into 10 raised to 6 Daltons, and that is the holoribosome. And when it gets dissociated into small subunit and the large subunit, that is the 50s and 30s, the 50s has got that is the 30s has got a molecular weight of about 0.9 into 10 raised to 6 Dalton. And if you take this particular large subunit of the ribosome, it has got a molecular weight of about 1.8 into 10 raised to 6 Daltons will be there. And this is some of the important information related to this particular prokaryotic ribosome. Now we'll move to the next part, and that is the eukaryotic ribosome. And in case of the eukaryotic ribosome, same thing is there, that is the holoribosome will be there. It gets dissociated into what we call it as a small subunit of the ribosome and the large subunit of the ribosome. And if you take the holoribosome, it has got a molecular weight of about 4.2 into 10 raised to 6 Daltons are there. And if you take this particular, once it gets dissociated into large subunit and the small subunit, the large subunit has got a molecular weight of about 2.8 into 10 raised to 6 Dalton. And if you take the small subunit of the ribosome, it has got a molecular weight of about 1.4 into 10 raised to 6 Dalton. These are the some of the important figures which are related to this particular holoribosome, small subunit of the ribosome and large subunit of the ribosome. Now we will try to discuss about the what are the different type of the components which are responsible for the formation of the ribosome. Now if you take this particular large subunit of the ribosome, first we will discuss about the large subunit, that is we are going to discuss about this particular thing. If you take this particular large subunit of the ribosome, we will find that there are 3 different types of the RNAs are present. In case of the eukaryotic prokaryotic ribosome, only 2 types of the RNAs are present, that is the 5s RNA and second one is the 23s RNA. But in case of the eukaryotes, you can find that there is an additional RNA is present, and that particular RNA is called the 5.8s RNA. That's why if you take the different types of the RNAs, in case of the last subunit, you can find that there is a 5s RNA is present. This is the first type of the RNA. The second type of the RNA is called the 5.8s RNA. This is the second RNA. And the third type of the RNA, larger than the what we have discussed for the prokaryotic ribosome, that is the, in case of the prokaryotic it was 23s, but in case of the eukaryotic you will get the 20s RNA will be there. And if you take the size of this particular 5s RNA, it has got a 120 bases are present. And if you take the 5.8s RNA, it has got 160 bases are present. And if you take this particular 20s RNA, it has got somewhere about 4,200 bases, 4,700 bases are present. And these are the information about the RNA. And now if you take the proteins, there are 49 different types of the proteins. It means that the eukaryotic ribosome is much more complicated as compared to the prokaryotic ribosome. Now we will move to the next this thing, that is the eukaryotic ribosome. And in case of eukaryotic ribosome, we'll find that this is the small subunit of the ribosome. And in this small subunit of ribosome, we can find that if you take it has got 40S, and if you take the 40S, you will find that there are only one. If you take the small subunit of the ribosome, you will find that only one type of the that is the RNA is present, that is called the 18S. Whereas in case of the prokaryote, we had the we have found that they used to have a 16S RNA, but in case of the eukaryote, you will find that there is a 18S RNA is present, and 18S RNA has got somewhere about 1900 nucleotides are present, and there are totally 33 different types of the proteins are present. This is the some of the basic details regarding the ribosome, because ribosomes are important because they are the sites where exactly the protein biosynthesis will take place, and that's why they are very important. Okay. Next, we will discuss about the enzyme. The last component of protein biosynthesis is the enzyme which is responsible for attaching the amino acid to the tRNA, because the tRNA should carry that particular amino acid to the site of the protein biosynthesis. That's why there is an enzyme. It is a catalyzed by means of a one particular enzyme. That particular enzyme is called the amino acyl tRNA synthetase. Basic things we already discussed. Synthetase means this is the enzyme which requires ATP or GTP for the energy, and this particular enzyme is nothing but this is the enzyme which is responsible for attaching amino acid to the tRNA. Again, I am repeating, this is the enzyme which is responsible for attaching the amino acid to the tRNA, and this particular enzyme is called the amino acyl tRNA synthetase. There are two different types of the amino acyl tRNA synthetase. One is called the class 1 amino acyl tRNA synthetase, and second one is called the class 2 amino acyl tRNA synthetase. There are two. Why this particular classification is required? Because they differ in their primary structure and they differ in their tertiary structure, but both of them are responsible for the same reaction. Okay, some alterations are there where exactly they attach the amino acid will differ, but one the important thing is that if you take these particular two the amino acid tRNA synthesis, you will find that they differ from each other in their primary structure, they differ from each other in their tertiary structure, and because of this particular difference, you can find that they are classified as class 1 amino acyl tRNA synthetase, and they are called the second one is called the class 2 amino acyl tRNA synthetase. And they not only differ in their structure, they also differ in the reaction mechanism. The purpose is same. What is the purpose? To attach the amino acid to the tRNA molecule. That is the actual process to happen. And how they accomplish this? If you take the amino acyl class 1 amino acyl tRNA synthetase, it will attach the amino acid to the second hydroxyl group. Okay, this is the second. It will attach to the second hydroxyl group. And what is the where exactly the second class 2 amino acyl tRNA synthesis will attach? They don't attach the amino acid to the second hydroxyl group. Instead, they will attach the amino acid to the third hydroxyl group. That is also very important. In this way, you can find that whatever these two different classes are there, class 1 amino acyl tRNA synthetase and class 2 amino acyl tRNA synthetase, they not only differ from each other in their primary structure and the tertiary structure, they differ in their function. And what is the difference? The difference is where exactly they are going to attach the amino acid. The class 1 amino acyl tRNA synthetase will attach the amino acid at the third hydroxyl group of the adenine, that is the CAA. In the adenine of the CAA, the second hydroxyl group of the adenine is the place where exactly the amino acid will be attached. Whereas in case of the class 2 amino acyl tRNA synthesis, you can find that in the CCA nucleotide, the bases in the adenine, there is a third hydroxyl group is present, and to the third hydroxyl group, that particular amino acid is going to be attached. That's why the reaction mechanism is slightly different, and this is some of the important things which we have discussed. I think we have covered some of the basic components. In the next video, we are going to discuss in detail about how exactly the protein biosynthesis will take place, and as a prerequisite to that particular understanding or the video, we have taken this particular video exclusively for to discuss about the different type of the components of the different types of components which are essential for the protein biosynthesis that we have discussed in this video. I hope this particular video is useful to you, and I think it's not so complicated. Only thing is that you should understand some basic things. What are the basic things related to the RNAs? What is mRNA? What is RNA? What is tRNA? This you have to understand. Then you have to understand what are the ribosomes and what are the importance of the ribosomes, where exactly the protein biosynthesis will take place, and what is the amino acyl tRNA synthetase, What is the function of the amino acid tRNA? Very simple. The amino acid will get attached to the tRNA, and this is a complicated mechanism. And in order to facilitate that particular process, there is an enzyme, and that particular enzyme is called the amino acid tRNA synthesis. It will facilitate. In this way, you can find that these are some of the basic components which are essential to understand the next video. I hope this is very much useful to you. And for this video and for this particular study material, I use some of the books. That is the Molecular Biology written by Bruce Albert, and Principles of Genetics, Snoostad and Simon, and Principles of Biochemistry by Nelson and Cox. I hope students should read these books, because hearing a lecture may give some help to you, but it is not going to give any knowledge. That's why, if you want to succeed in your life, develop the habit of reading books. That is my sincere advice to all the students. Thank you very much for observing this video. With this, we are going to stop, and in the next video we are going to discuss about the exact process by which the protein biosynthesis will take place. We are going to discuss it in molecular details, whatever the different type of the factors are responsible, whatever the different type of enzymes are there, and whatever this particular process there, it will be divided into different stages. And by taking different stages, we are going to understand the protein biosynthesis. That is going to be there in the next class. I hope you will be able to observe that particular video. Thank you very much at this point of time.